everyone. Thank you, first of all, to the organizers for giving me this opportunity to present my work. So, as mentioned, I'm part of the U1118 in Strasbourg, and I'll present today some of my PhD work looking into variants in NUP50 as risk factors for ALS. So NUP50, or nucleoporoid 50, was one of the hits in a TWAS performed by a researcher in our lab, done on the largest GWAS dataset of ALS patients. And what is NUP50? It's one of the many nucleoporins that form the nucleopore complex, which is essentially a channel in the nuclear envelope between the nucleus and the cytoplasm that is involved in the shuttling of proteins and RNAs between these two compartments. And this is interesting to us as it is a function that is suspected to be affected in ALS. As you know, there is the cytoplasmic mislocalization of normally nuclear proteins such as TDP43 and FUS. A mounting evidence over the last, let's say, 8 years has pinpointed that some nuclear pore components are mislocalized in ALS. This is the case in various models of ALS, such as in cellular models of TDP43, also in C9ALS, in postmortem brain tissues, as well as in cellular models. And this was also the case of NUP50 to be aggregated in SOD1 ALS mouse models and in sporadic ALS tissue. Furthermore, one of the SNPs identified in NUP50, which is quite common, led to a decrease in NUP50 in the dorsal lateral prefrontal cortex, and is considered as an ALS risk factor. And knocking it down in cellular models led to an increase in cellular death, and knocking it down in animal models, such as in zebrafish, led to swimming defects, which could be rescued by reintroducing the human form of NUP50, making it all consistent with an ALS-like phenotype. Furthermore, rare variants in NUP50 have also been identified and are predicted to be pathogenic. They are mostly localized in the important alpha binding domains and NUP153 binding domain. But how they lead to alterations in the NUP50 function has not been studied yet. So the straightforward question of my talk today will be looking into what are the underlying mechanisms of the pathogenicity of these variants. And we have generated multiple NUP50 mutant plasmids, all tagged with an HA tag. And I'll first talk about their localization. So we use the HT22 cell line, which is a murine hippocampal-like cell line, to look at the localization of NUP50 within the nucleus. NUP50 is actually one of the nucleoporins that is not only at the nuclear pore, but shuttles between the nucleoplasm and the nuclear pore. So that's why we also looked at its localization within the nucleus. And we didn't find any difference between the wild type or the variant plasmids. So they appear to localize normally. And we also further confirmed this by using a marker of the nuclear pore complex. And all these variants and the wild type normally localized in the nuclear pore. So far the variants have a normal localization, but we wanted to study also their interactome. So we took an example of a variant localized in the important alpha binding domain and another one in the NUP153 binding domain. And we used the HT22 cell line as a neuronal-like cell line and the hex cells as a non-neuronal cell line. We pulled down the proteins linked to the variants with anti-HA beads and analyzed them via mass spectrometry. On the silver staining, we see subtle differences in bands. And when we look at the interactome of NUP50, the wild type, we can see that we have different data sets for them, first of all, to compare. We have the top data sets that are ours, and then the knock-in model from our collaborators in Strasbourg. And we can see that NUP50 does interact with nucleoporins, importance, FUS, TDP43, and SWI-SNF subunits. And this is reflected in KEGG analysis, where the major function of the interactors of NUP50 is nucleocytoplasmic transport, but it is also ribosome and spliceosome, which was unexpected and needs to be further investigated. When we compare the variants compared to the wild type in terms of their interactome, we can see that the R45C variant loses its interaction with all important alpha binding partners, but this is not the case with the other variant, Y156C, so it is specific to this variant, and this is also confirmed by Western blotting. Looking at the Y156C variant, we can see that it loses its interaction by around half compared to wild type with NUP153, which was not seen with the other variant, and is further confirmed by Western blotting. So these variants that we have studied so far have an altered interactome, and we wanted to see whether they led to motor defects in an animal model. So with the help of our collaborators at the Institut Imagine, we injected these variants in zebrafish, and we can see that there was no difference in phenotypic distribution between wild type and mutants, although the overexpression of the wild type also has some slight effect compared to non-injected zebrafish to keep in mind. And looking at the motor test, we can see that the variants R45C and Y156C led to a decrease in distance swam compared to wild type and non-injected mice, zebrafish, sorry. And looking into the motor neuron morphology, we can see that these variants also had defects in axonal branching compared to non-injected zebrafish, and that there was a decrease in motor neuron numbers with the Y156C variant. So today, in conclusion, I will have shown that in cellular models, the NUP50 variants have a normal localization at the nucleopore and within the nucleoplasm, that NUP50 normally is related to nucleocytoplasmic transport ribosome and spliceosome, and that the alterations that we have studied in the variants are mutant-specific. And they seem so far to be more loss-of-function variants, but we cannot exclude that there are gain-of-toxicity mechanisms that we haven't identified yet, especially looking at the motor deficits we observe in zebrafish models. So I'd like to thank all the team working on the project, our collaborators, ARSLA for funding, and thank you for your attention.
Thank you, Olga. Very nice presentation. Do you have some questions? Séverine?
Thanks, Olga, for the presentation. So you actually show that you have one nice phenotype with one variant. And so maybe I missed it, but this is supposed, because you talk about the loss of function, this would be supposed to lead to a loss of function and to make a stop or not. And my second question is, wouldn't you expect actually that in those, I mean, if we want NUP50 to be a cause of ALS and therefore to touch the motor neurons, actually, wouldn't you expect that you wouldn't see much in the other cell lines and you would have to look for the localization more in motor neurons, and that could be, indeed, you could see a difference.
Definitely, this could explain why we don't see things in our cell lines, but we are planning to do differentiated neurons from iPSC, so we'll check. But at least from data prior, we can see that it doesn't seem to change too much, but maybe the variants would have an effect in motor neurons rather than cell lines. And then for the Y156C variant, so it does lose its interaction with NUP153. So this maybe could explain why we see motor neuron loss, at least in the zebrafish model. But it doesn't give the full picture because, I mean, we still need to check, but it's definitely maybe both loss of function and gain of toxicity. And also NUP50 I haven't mentioned, but is involved in development. So maybe that's why we see a stronger phenotype when we inject it in zebrafish during development.
Thank you.
Okay.
Suzy.
Small question about the fact that you switch, you have your two variants, okay?
Yes.
They both switch to a cysteine. Can you comment on the mechanism that can be involved? Have you checked whether you get other disulfide divide the bridge and stuff like this. Can you comment?
The R45C is considered as a structural variant, so the cysteine will definitely change the conformation of the NUP50 protein, but the Y156C is not considered as a structural variant, so it's, I think, mostly due to the loss of interaction with NUP153, but yes, we haven't checked at the conformation of the proteins itself. It's more in silico predictions.